us an objection. So in this talk, I argued that the ascription of normal functions to parts of cancers can be explicated by a novel account, which I call the modeling account of function. The modeling account is a modified organizational account of function of the sort put forward by Matteo Massio, Christian Sabarito, and Alvaro Moreno in their 2009 BJPS paper. First, so I chose them because they have sort of done the most work in providing an organizational account and then sort of extending it. They have like a ton of papers in the last 10 years sort of extending this account, so I draw from it. So first, I claim that any account of function committed to explicating ascription in a given subdiscipline of biology, which I call descriptive accounts, must satisfy two desiderata. I call these desiderata class adequacy and explanatory adequacy. Second, I present a representative example of cancer biologists ascribing a normal function to a part of a cancer. I then present the modeling account of function, which claims that the type of system to which functions are ascribed in cancer biology are self-organizing systems, which I will define later in the talk, and that candidate activities for ascription of normal functions are identified by modeling disease progression. I conclude by considering an objection to the claim that cancer biologists ascribe normal functions to parts of cancers. Okay. So function ascription is pervasive in biology. To a first approximation, functions are activities that parts of biological systems perform, and that by being so performed, contribute in some way to those systems. So the favorite philosopher's example, the function of the heart is to pump blood. This ascription tells us that hearts pump blood, or that they should, and that pumping blood contributes in some way to biological systems with hearts. For instance, by transporting nutrients and waste to and from various tissues in the body. Ascribing a function explains by drawing our attention to the dispositions and structural features of systems that are or are supposed to be causally relevant for system-level phenomena of interest. Biologists are keen to understand how and why biological systems persist and propagate. Functions are indicative of how those systems do so, or why they have the dispositions and features which, in the good case at least, allow them to do so. So different types of function are ascribed in different subdisciplines of biology. For instance, in cladistic systematics, functions are ascribed to homologous traits to acknowledge the continuation of those traits from a common ancestor, or novelty in them or their activity. So the carapace of Proganocalis, a genus of prototurtle, is ascribed the function of protection and recognition of its novelty as a trait. Importantly, this ascription tells us what causal role the carapace played, not what it is supposed to do. There are no more Proganocalis, and the cladis is not committed to the claim that the top part of the shell, which is what the carapace is, had the normal proper function of protection. By contrast, physiology is the branch of human biology that is focused on so-called normal proper function. The heart's pumping is a case in point. Physiologists and doctors regularly appeal to it despite knowing that many hearts fail to pump blood. The position that distinct types of function are ascribed in distinct subdisciplines of biology is called function pluralism. And it's been widely adopted among philosophers of biology. Everyone is a pluralist about function nowadays. However, I'll be concerned primarily with normal functions throughout the rest of the talk, especially as ascribed to parts of cancer by cancer biologists. These functions are standard for part activity, disposition, and structure. So the heart's function embodies a standard for its pumping, its disposition to pump, and the structure that allows it to pump. I'll also restrict focus to accounts of function in the philosophy of biology that aim to explicate the ascription of function in and across subdisciplines of biology. By contrast, accounts like Ruth Millikan's account of proper function aim to tell us not what biologists mean when they use function talk, but rather attempt to provide a definition of function that biologists they think should take up, or which might make their usage more consistent. These are often called prescriptive, though I don't necessarily want to conflate Milligan's account with other prescriptive accounts of function. In restricting focus, then, I restrict focus to descriptive accounts, or what I call descriptive accounts. And there are at least two desiderata that I think these accounts have to satisfy. So the first is class adequacy. And I take this to be a more ontological desideratum. Accounts should be consistent with the subdisciplines of biology they attempt to explicate concerning the class of systems to which functions are ascribed. And I draw this from the thought that many critiques of accounts in the literature are based on showing that they imply either that some system which shouldn't be ascribed a function has a part which that account allows for the ascription of a function to, or it fails to capture a system, it fails to apply to a system to which it seems either intuitive that it should, or in which the biologists in question actually do ascribe such functions to. So this would be being too liberal or too narrow, respectively, too broad or too narrow, respectively. So I have a couple of examples here. One is if you have an account of normal function, your account is too broad if it allows you to ascribe normal functions to parts of the solar system. It's too narrow if it doesn't allow you to apply normal function descriptions to, or ascribe normal functions to organisms or possibly ecosystems. So the second desideratum is less ontological and more epistemic. In this case, accounts should be consistent with the subdisciplines of biology they attempt to explicate concerning the principles that guide, either implicitly or explicitly, the ascription of function. So for instance, Ronan Munson, George Lauder, and Paul Griffiths have argued that cladistic systematics don't appeal implicitly or explicitly to the actual evolutionary history of a trait in order to identify it as a homologue. If they're right, functions ascribed in cladistic systematics are not etiological or historical. If so, then Karen Neander's account of normal function, which is a selected effects account, an etiological account, is not descriptive of cladistic systematics. Of course, she disagrees, right? There's this dialectic that they engage in the 90s and early 2000s. But that aside, I think just the dialectic itself suggests that the explanatory adequacy is a legitimate desideratum on descriptive accounts. So I take it that meeting these two desiderata, class adequacy and explanatory adequacy, constitute a minimal hurdle for descriptive accounts relative to some subdiscipline of biology. All right, so now on to the example. So a consistent challenge for those working in cancer biology is dealing with treatment-relevant variation among cancers. The following is a non-exhaustive list of dimensions of treatment-relevant differences that individual cancers can exhibit. So you have anatomical site and tissue type of origin, genome, mutation rate, growth rate, tumor formation, incidence and rate of metastasis, the cancer microenvironment, and initiation. And within initiation, you have things like environmental carcinogens, pathogens, genetic diseases, and so on. So that's at least 9 dimensions on which individual cancers can just differ from one another. And that's non-exhaustive. So like inquiry in any domain, a central task in cancer biology is finding within all of this variation, sameness that's of causal and explanatory relevance. For instance, cancers have historically been classified by anatomical site, tissue type, stage, and grade. A stage 1, grade 1 lung adenocarcinoma is a cancer originating in glandular tissue or glands, that's the tissue type in the lung, the anatomical site, that has yet to form a tumor stage and whose cells still resemble healthy somatic cells, grade. So the standard classificatory scheme is effective at grouping cancers together and bears explanatory fruit. For instance, other properties relevant for treatment often cluster around tissue type, stage, and grade. Only some types of tissue form solid tumors, that is clumps of cancer-associated cells. Size is a property of solid tumors that is predictive of disease progression. So you can't really infer disease progression in this case unless you're assuming that we're dealing with a solid tumor or solid cancer, right? So this sort of inference about size being predictive of disease progression doesn't work for liquid cancers like leukemias. Another example, the degree of apparent similarity between cancer cells and healthy cells is also predictive of growth rate and metastatic potential. So grade 4 cancers with cells that are very unlike their healthy kin are likely to grow and metastasize more quickly and aggressively. However, the standard classificatory scheme is not perfect. So for instance, cancers originating in the same organ can be genetically more similar to those originating they can be more similar to those originating in a different organ than to each other. Precision oncology depends on targeting specific mutated genes and proteins. So sameness in anatomical site of origin is not always explanatory or helpful for guiding treatment. Luckily, this scheme presents only one of many tools for finding treatment-relevant sameness among cancers. One tool that cancer research shares with several subdisciplines of biology is ascribing normal functions, or so I'll now argue by example. Okay. So in a widely cited paper, Peinado et al. 2012, claims to have, quote, explored the function of melanoma-derived small extracellular vesicles, or what I abbreviated as SEV, in the formation of primary tumors and metastases. Zhang and Yu, 2019, reporting their results say, quote, Peinado et al. 2012, I've added that, have advanced our understanding of the novel function of SEV in pre-metastatic niches. The novel function explored, and of which our understanding is advanced, is the delivery of a signaling protein to cells in bone marrow via small membrane-bound packages produced mostly by advanced melanomas. So this is what I have in the figure. Through a series of experiments, Peinado et al. Identify an activity that melanoma-derived SEV perform, and which results in greater primary tumor growth and more aggressive metastases. Melanomas produce SEV carrying mesenchymal epithelial transition factor, an oncoprotein that can trigger several signaling pathways in cells. Melanoma-derived SEV carrying MET, which is what I abbreviate mesenchymal epithelial transition factor as, travel through the blood to cells deep in bone marrow that have yet to differentiate. Receiving MET sets off a cascade of signaling in those cells that mobilize them to inflame distant organs, as well as cause those organs to exhibit vascular leakiness and produce vascular tissue. The result is pre-metastatic niche formation, which promotes greater primary tumor growth and metastasis. So I think Peinado et al. Features the ascription of a normal function. Beyond use of the definite article, both Peinado et al.'s and Zhang and Yu's talk of the novel function of melanoma-derived SEV generalizes over them without distinguishing between stages of disease progression, SEV that successfully deliver MET, SEV that are deformed or fail to carry MET, or melanomas that fail to produce any SEV at all. So they just generalize over all of these differences. And I think that's not an accident. So in the process of identifying the function, Peinado et al. Examined SEV production and MET delivery across melanoma patients at distinct stages of disease progression, as well as low and high metastatic melanoma mouse models. These are cases in which they model the cancer both by having mice into which they inject the cancer and mice which they grow the cancer in initially. They also examined SEV production in melanoma mouse models designed to produce SEV lacking MET, fewer SEV, or no SEV. The point was to home in on mechanisms responsible for SEV-mediated pre-metastatic niche formation. This in turn required Peinado et al. To re-identify SEV or mark their absence, and to identify and relate in a systematic way the effects of their presence or absence on niche formation. Some of this was accomplished by tracking SEV-related proteins in blood. However, at least some of it was accomplished by hypothesizing the activity melanoma-derived SEV are supposed to perform. positing the dispositions and structure that in the good case for the cancer allow them to perform it. Their hypothesis drew Pinato et al. To look for SEV and MET in bone marrow and potential sites of metastasis in patients and mouse models. It also drew them to infer from a lack of SEV, a lack of MET, reduced tumor growth and reduced metastasis, that they had successfully disrupted the functional dispositions or structure of melanoma-derived SEV. In confirming their hypothesis, Pinato et al. Showed that deformed SEV and SEV that don't carry MET are in some minimal sense supposed to deliver MET to bone marrow and are in some minimal sense supposed to have the dispositions and structure that allow them to do so. I will elaborate in a moment what I think that amounts to, but for now, the generalization over both defective and non-defective SEV in the process of discovering their function and possible clinical interventions suggests that the ascription identifies a standard for part activity, disposition, and structure. In which case, I argue, delivering MET to bone marrow is a normal function of melanoma-derived SEV. Okay, so that's the example. I'm hoping I have convinced you that it's an example of cancer biologists ascribing a normal function to a part of a cancer. So now my positive account. So far I've claimed that different types of functions are ascribed in and across subdisciplines of biology, that adequate descriptive accounts of function must meet class adequacy and explanatory adequacy, and that cancer biologists ascribe normal functions by means of example. I don't have the space in this talk to contrast the modeling account with its rivals, although I'm happy to discuss how I think it does better than them in the Q&A. For now, I want to argue that the modeling account satisfies both desiderata with respect to the ascription of normal functions to parts of cancers in cancer biology. As something of a preview, the modeling account finds application in cancer biology. However, in future work, I hope to show that it is descriptive of ecology, cladistics, and physiology, as well as some subdisciplines of evolutionary biology. So the first desideratum I think is satisfied by focus on the class. So the class of systems the modeling account posits lines up with the standard classificatory scheme in cancer biology, but can be extended. Finally, the modeling account prioritizes modeling as a way of establishing standards for part activity, disposition, and structure. However, other sources of evidence can confirm or disconfirm function ascriptions. For instance, histological or genomic evidence. All right, so the modeling account satisfies class adequacy by adopting the class set forward by systems biology and organizational accounts of function, namely organized self-maintaining systems. So I think of a system as organized in the sense that the parts composing it are arranged into, in principle, decomposable functional units at multiple levels. So the cardiovascular system is a functional unit that is distinguished from the nervous system, which is distinguished from the skeletal system, the muscular system, the endocrine system, and so on, in part by being a system which transports nutrients and waste around the body. The heart is distinguished from arteries, veins, and the like by being a pump, and so on. I think of these as self-maintaining systems in the sense that the activities of the parts set up the conditions for those very activities by keeping the system organized as it is. So the pumping of the heart sets up the conditions for future pumping of that heart, in part by transporting nutrients and waste, or aiding in the transport of nutrients and waste, which keeps the system organized as it is, so that pumping continues to do its job, basically. So the reasoning behind this choice is that cancers are not organisms, but they're not quite not organisms either. And I have a similar bit of reasoning for ecosystems. However, it differs in the following way. Cancers are arguably atavistic, returning to a point in evolution where multicellularity had not yet established itself, and cooperative collections of cells were still not fully integrated into autopoietic wholes. Cancers are also not single-celled organisms, however, but they are pathologies, that is, breakdowns in organismal health or healthy functioning. So philosophers who champion naturalistic accounts of health often define pathologies as a loss of health. Ontologically, pathologies are processes and parts of an organism, not entities unto themselves. Yet cancers are both disease processes, there are processes, and entities unto themselves. This double life requires cancer biologists to engage in doublethink about them. The class of organized self-maintaining systems provides a promising way for them to do so, as such systems can be understood as in terms of processes, this is how systems biologists understand them, and as comprising further systems, for instance, organisms in an ecosystem. All right. So the modeling account satisfies explanatory adequacy by appeal to modeling. I take a very broad view of modeling following Godfrey Smith and Levi. So I just think of models as representations that abstract or idealize features of what they represent with a view to predicting or explaining the behavior of their targets in much the same way that we infer the location of a place by way of locating it on a map. I also don't necessarily want to be committal on the resemblance or similarity relation that is supposed to obtain between models and their targets, except insofar as that I think that the modeling account aims to be comparative. So on the account, when a normal function is ascribed to a part, there is some representation such that for any two models of the system, if one model is to include that representation and one were to lack it, the first would better predict or explain the contribution made by the part's activity. So I think if you had two models of the cardiovascular system, one that includes a representation of the heart is going to better explain the transportation of nutrients than one that doesn't. That's the intuition. Moreover, Peinot et al. Relied heavily on modeling to identify the normal function of melanoma-derived SCV, to bring us back to our example. Modeling is an indispensable source of evidence in cancer biology, and much of biology besides. And in Peinot et al.'s case, you can see that every link in the inferential chain to their function description is forged using both human cell line models and mouse models. It's especially fruitful, modeling that is, by helping us home in on mechanisms. In this case, the mechanism of SEV-mediated pre-metastatic niche formation is elaborated by the identification that it is the delivery of MET to bone marrow that facilitates niche construction. Since cancers are highly variable, finding such mechanisms is extremely difficult without some way of setting a standard. Modeling allows us to set such a standard, and this is the key lesson that I want to draw on in presenting the modeling account. Other accounts of normal function rely on the normalizing effect of selection or on conditional statistical typicality to set a standard. So I'm thinking of Neander, Millikan, and Borse, and Piccinini, and Hausman, and others. The modeling account, by contrast, appeals to a novel way of establishing a standard, one that's admittedly more thoroughly epistemic, namely whether we can create explanatory and predictive models of the contribution the function-bearing part provides by performing its function. I don't think that this makes the account interest relative in the way some philosophers of biology tend to worry about, and I can explain more why I don't take that to be the case in Q&A. All right, so because we started a little late, I'm gonna maybe push a couple minutes, but I do want to address this objection. So there's at least one objection to the claim that cancer biologists ascribe normal functions to parts of cancers. The objection amounts to this. There's no reason to think that the ascription of the function or the novel function to a part of a cancer identifies a standard for its activity, dispositions, or structural features. That these phrases show up in cancer biology is likely just loose talk. These functions are no more than the actual or typical causes of disease progression, for instance, metastasis. Talk of function signals only that these activities are functions of the kind described in, for instance, statistic systematics. Moreover, cancer biologists reserve talk of, quote, normal functioning for the healthy variants of the disease processes they study. So this is sort of always lurking in the background, right? In response, there is at least one reason to try to identify standards for part activity, dispositions, and structural features among types of cancers. Namely, doing so successfully provides viable avenues for treatment. The ultimate aim of cancer biology is the development of effective clinical interventions. This aim is problematized by the variability that cancers exhibit, which I discussed sort of the beginning of the example section of the talk. And it's further problematized by cancers effectively exploiting that variability. In particular, cancers that escape immune response and resist treatment are more likely to effectively maintain themselves, at least until the patient's death. The more thoroughly a cancer establishes itself in its host, the more specialized the knowledge required to root it out becomes, especially if it becomes entrenched in part in light of attempts at treatment. At the same time, the variability that a cancer can exhibit and exploit is constrained by the evolutionary history of the organism, the mutations driving the cancer, the rate at which the cancer mutates, the organism's environment, and several other things, so more things that I'm not going to get into. These limits on variability can guide cancer biologists to commonalities across cancers, hence the use of a plurality of classificatory schemes. I submit that knowledge how to undermine particular traits or mechanisms of types of cancers is often gained by identifying their normal functions. Identifying these activities as standard cuts across instances in which the relevant traits or mechanisms fail to behave as predicted. They are abnormality inclusive. Idiosyncratic failures may not be a focus of cancer biology because clinicians may not want to rectify those failures for obvious reasons. Nonetheless, in seeking to undermine the disease, cancer biologists identify standards for those activities, dispositions, and structural features of parts of cancers that many cancers do actually embody. That is, in seeking to undermine the disease, cancer biologists try to figure out how those cancers work. So I want to stress that giving up on normal functions here threatens to deprive us of an extremely useful tool in the cancer biologist's toolbox. I also want to grant that cancer biologists sometimes speak in confused ways. Even if their ascription is a dispensable part of cancer biology, the case presented by Pinato et al. 2012 shows us that normal functions serve as an effective guide to at least some commonalities of clinical significance among cancers. The resistance to allowing normal functions to be ascribed to parts of cancers rests, I suspect, in the conflation of what something is supposed to do with what is healthy. This conflation is exacerbated by the fact that cancer biologists will often refer to the activities of healthy variants as their normal functions, reserving, quote, the function, quote, the novel function, quote, the novel function of X in cancer, or, quote, the normal function of X in cancer, unquote, for the ascription of normal functions to parts of cancers. Despite appearances, I believe all are tracking what I take to be normal functions. To back this up, I should, in conclusion, I am going to disambiguate normality from health in a very standard way. So I think health is a state that is good to be in, and what is healthy is conducive to being in that state, whatever it is. The heart's pumping efficiently is healthy. But health is not synonymous with normality, where normality merely sets a standard for a type of system against force, obviously. Consider an Olympic sprinter whose leg muscles are quickly atrophying. The state of her leg muscles is unhealthy, despite crossing what is normal for the species on a number of dimensions, for instance, volume or mass, as they wither. By contrast, an Olympic sprinter in their prime will have leg muscles that differ from what is normal on those dimensions and many besides, right? So an Olympic sprinter is going to have much more massive calves than what is normal. Normality in health can therefore come apart, force notwithstanding. Normality sets a standard, and some parts of cancers have standard activities, dispositions, and structural features. That cancers are pathological, and a cancer biologist will often talk of normal functioning when they mean or should say healthy functioning in no way undermines the legitimacy of ascribing normal functions to their parts. So in summary, different types of functions are ascribed across biology. Descriptive accounts of functions relative to those subdisciplines or across them must satisfy class adequacy and explanatory adequacy as minimal desiderata. Cancer biology features the ascription of normal or abnormality inclusive functions. And my account, the modeling account, provides a promising descriptive account for understanding this ascription by satisfying class and explanatory adequacy relative to cancer biology. All right, thank you so much.
Okay, so we have time for questions. Are there any questions for Seth?
And sorry if the audio wasn't too good. Being outside makes it a little bit difficult.
Any questions?
Have I convinced you all?
Dr. Potoshnik.
Thanks for the nice talk. Simple question, but something that occurred to me as you were walking through the possible objection that effectively that sort of function is only being used metaphorically in cancer biology contexts. I found your response really compelling that it's doing actual work there, but sort of the objection and response made me wonder, and this is an outsider question because I don't think about functions, but what do you think is at stake in the question of whether function is intended literally or metaphorically in any given context? So it struck me as sort of, like I said, I found your response to the objection compelling, but like I also sort of at the end was like, well, if it's doing work, what is there beyond that that is supposed to be involved in ascribing functions in any of these other contexts?
Thank you. Yeah. So I think what the work that it does is mostly explanatory. Well, explanatory and predictive. Explanatory in the sense that it explains the mechanism of, for instance, pre-metastatic niche formation, or more specifically, small extracellular vesicle mediated pre-metastatic niche formation, right? So you want to understand why, for instance, the lungs are inflamed, why they're vascularly leaky, why they're growing vascular tissue. You can explain it by appeal to the function of these small extracellular vesicles and what they're doing. And you can also predict from looking at patients who are producing a lot of these vesicles that they're going to be subject to metastasis. They're gonna experience metastasis. So that's like the work that the ascription of the function is supposed to do. What's at stake is using them in that way for the sake of clinical intervention. So if we just were to say, okay, function talk is purely metaphorical, it doesn't really tell us anything literal about what's going on. It doesn't give us a standard for these traits. It's just sort of a way of speaking. I think you threaten to miss how these explanations are supposed to help you develop treatments for patients. So realizing that the function of these vesicles is to carry an oncoprotein to bone marrow cells, like taking that literally allows you to quickly come up with a good clinical solution, assuming you can, if you can pan it out, right? Which is to stop them from carrying that oncoprotein or stop the production of that oncoprotein, right? And in fact, this is what peinatoidal do, right? They create melanomas that are unable to produce this oncoprotein, and they find that the mouse models don't exhibit metastasis, right? So what's at stake is like better clinical interventions, basically, or just even the possibility of clinical interventions in some cases. And thank you for that question. That's actually a very good question.
Any other questions for Seth?
I'm also happy to walk through things that I sort of tabled if people
Bill
have
Harper,
questions.
Bill, you can ask your question.
What if this is, in a certain sense, the question you just got is the one I was going to ask. And I just want to, it seems pretty clear that from what you've said, that looking at the cancer in a body as an organization trying to do certain functions really does help understand what would be the more useful things to do in order to help the person with the cancer survive or be less badly affected. I'm surprised that people don't just say, "Of course you're right."
Well, thank you. Yeah, I have similar intuitions. However, so Justin Garson, who has a generalized selected effects account, a function, wants to allow that cancers have function. There's a 2017 paper of his where he's arguing against organizational accounts of function. And he, as sort of an aside, he says, well, if you think of these systems in this way, then yeah, they have functions. And Hausman has, whose first name I don't know, has a 2012 paper on functional efficiency where he notes that people talk this way and he tries to give some legitimacy to that sort of talk. So I'm not necessarily arguing against them. However, I think I'm arguing against more of what maybe I would call the old guard of functionalist philosophers. So I'm thinking of Karen Neander and Christopher Borse, who in various of their works would say as asides, like, well, of course, no pathology has a function. And of course, like cancer, because it's a pathology, can't have any functions. Borse, I think, says this in a couple of places in either 2001 or 1991 long paper of his, where he says something like this in a couple footnotes. So I'm mostly giving the example for people of that persuasion who may be hardcore Neanderians or Borsians. And then the sort of second move that I want to make is to say that extant accounts, which I don't do in this talk, but I do in the paper, extant accounts like the biostatistical account and the selected effects account don't do as good a job as my account.
I think focusing on the advantages that are coming at this way of approaching it would be a really important thing to do.
Yeah, I mean, so, and I can walk through that if people want, if there aren't other questions. It's up to, I think, I'm happy to go with whatever you guys want.
Yeah, feel free to elaborate.
Yeah, so I contrast my view with three accounts. One is the organizational account that I draw from. And the reason I contrast my view with theirs is because their view is not, in the first instance, an account of normal function. So, and when they do talk about normal function, they have this sort of ad hoc way of trying to establish system types and then talk about tokens relative to those types. So I say, I try to create a dilemma for them and then just say, the better sort of solution is to, if for them at least, is to try to be a selected effects account effectively of normal function. So then the main criticisms are on the selected effects account and the biostatistical account. Against the selected effects account, I say, unsurprisingly, that there are parts of cancers to which functions are ascribed, but which don't seem to be selected for, which is a necessary condition on selected effects accounts. They argue that for something to have a normal function is for it to be selected to perform that function, right? So the heart has the function to pump blood because in our ancestors, right, way back, you had systems, you had vertebrates that had hearts and you had vertebrates that didn't have hearts. And the ones that had hearts won the Darwinian game, right? And this created a natural selection created a situation in which you had mostly vertebrates with hearts. And that selection pressure for hearts has continued in force for the rest of evolutionary history. So I try to argue by example that there are at least some parts of cancers which aren't selected for, which cancer biologists try to ascribe functions to. I'm not recalling the specific example. I can pull up the draft if I need to, but that's sort of the move, is to say, here's a counterexample. Against the biostatistical account, I raise very common criticism. So for the biostatistical account, for something to have a function, it has to be doing what it typically does, given that it's conditional on its contributing to the individual survival or reproduction of the individual organism. And typicality is defined relative to a reference class. So for us, it would be, the reference class would be human beings of a certain age group with a certain biological sex, a certain blood type, and so on, right? You try to fill out the reference class specification. Reference class specification is notoriously difficult. And I try to argue in the paper that it's way more difficult than it is in the regular biology case in cancer biology. In cancer biology, type individuating cancers is actually super hard. And there isn't really a clear way to do it. So I think the better solution is to give up on trying to come up with an ontologically rigid way of type individuating cancers and accept something like the standard classificatory scheme with its flaws and other such schemes, take type individuated cancers like melanomas and model them. And then you can add specifications as you go. And in that way, right, I think I improve on biostatistical accounts. So I get rid of the reference class issue by not appealing to typicality. I think appealing to typicality, which then requires a reference class, which then requires type individuation, is too difficult. And then for the selected effects accounts, I just say there are going to be instances where there are functional parts of cancers that just aren't selected for. However, I should say, so I should preface this a little deflationary, that in the paper itself, I do try to be more ecumenical. I say my view I think is the best. However, I think you can adjust things with these other views to try to do what I do. And there's some wiggle room, and maybe people who are diehard selected effects accounts proponents or statistical or biostatistical accounts proponents can dispute the examples I want to bring up as ones showing that their theory isn't good.
Any final questions for Seth? Dr. Sullivan?
Hi, Seth, I really enjoyed that. I wanted to ask you, I mean, cancer is kind of a unique case, but given sort of the broader context of health and normal function that you mentioned at the end, I'm wondering if this applies to other kinds of cases in medicine. If you had mentioned there were cases in ecology, but I was curious if cancer is like really unique in this way. Did you get
Thank
that?
you. Yeah, so in that 2017 Garson paper, one of the, I mean, his main sort of criticism of organizational accounts is that they ascribe normal functions or functions to self-maintaining pathologies. He provides cancer as an example, but then, says like maybe cancers do have functions. So, but the example that he draws on as a counterexample is panic disorders. So there are going to be instances in which panic functions to perpetuate panic, right? Anxiety disorders probably have a similar sort of structure. And there are probably, I mean, so this is more clinical psychology than medicine per se, but self-maintaining pathologies would be sort of the relevant category. And I would think that, and I haven't worked it out specifically, I haven't looked at the literature on panic disorders, and I haven't looked at this literature, so I can't actually say on firm ground really that I know that my account applies. But I would suspect that any pathology that is of the category of self-maintaining pathologies would be amenable to my account. Would be one to which my account applies, however you want to put it.
Tell me about your crisis of faith. Yeah, tell me more about that. You told me about your.
All right, so if that's all the questions, I think we can thank Seth once again. We'll take a break and we'll come back at 2:45.
Thank you so much, everyone.
Technology, and I really want to, but like, what about that gap period?